Welcome back to our biology class. Right, today we are going to study about carbon dioxide is essential for photosynthesis. So in the last class we studied about factors of photosynthesis. In factors of photosynthesis, there we studied how water is useful for photosynthesis, and whereas air and photosynthesis. Joseph Priestley experiments. Joseph Priestley, he found the oxygen, whereas oxygen is named by Lavoisier. So we discussed about two factors which are related to the photosynthesis, that is water and air. Then here we are going to see from the air carbon dioxide is essential for photosynthesis. We are going to prove that carbon dioxide is essential for photosynthesis. So this is practically proved by moles, half, moles scientist. That's why we can also call this moles half leaf experiment. Okay, ఈ ఎక్స్పెరిమెంట్ని ప్రాక్టికల్గా ఎవరు ప్రూవ్ చేసిందంటే మోల్స్ అనే సైంటిస్ట్ ప్రూవ్ చేసింది, కాబట్టి దీన్ని మోల్స్ హాఫ్ లీఫ్ ఎక్స్పెరిమెంట్ అని కూడా అంటారు, లేదా కార్బన్ డైఆక్సైడ్ ఫోటోసింథసిస్కి అవసరమని ఎలా నిరూపిస్తాం. సో దిస్ ఈజ్ వీఆర్ గోయింగ్ టు ప్రూవ్ దట్ కార్బన్ డైఆక్సైడ్ ఈజ్ ఎసెన్షియల్ ఫర్ photosynthesis. So this is an activity. This activity, okay, let us start with some basic steps which are aim, material, procedure, observation, conclusion. So these are the steps that we have to be followed. Okay, activity ఎప్పుడు చేసినా మనం ఇక్కడ ఈ aim, material, procedure, observation, conclusion అనే steps follow అవ్వాలా, right? So before the experiment, first we have to be selected one healthy potted plant. Then that healthy potted plant should be kept in the dark room to free from the starch, so that the plant must be de-starched. Before the experiment, the plant which is selected for this experiment has to be de-starched. For this purpose, we kept here this we have to be kept this plant in the dark room for few days, suppose for 3 to 4 days according to our textbooks, right? The potted plant which is selected for this experiment it has to be kept in the dark room for 3 to 4 days. Okay, after 3 to 4 days it must be taken out from the dark room and exposed in the presence of sunlight. This should be kept in the presence of sunlight. When it is kept in the presence of sunlight, here we should make some arrangements. What see, let us see. First, aim, the activity we are going to start here. Our aim is carbon dioxide is essential for photosynthesis. So the aim already I have written here, carbon dioxide is essential for photosynthesis. The next one, material. What material is needed for this experiment? One is potted plant. Okay, right. Next, transparent bottle, wide mouthed bottle. Transparent wide mouthed bottles. Here transparent means light should pass through this bottle, that is transparent bottle. And wide mouthed, this bottle should have splitted cork. See, this is splitted cork, right? Whereas this is transparent bottle. Okay, and next, potassium hydroxide solution, KOH, potassium hydroxide solution. And another one is iodine. And next, petri dish. So these are the things which are needed for this experiment. And procedure, another one is procedure. Procedure law, first we selected the potted plant, it is kept in the dark room for few days. Okay, then after few days, 3 to 4 days, again it has taken out from the dark room and kept in the presence of sunlight, and we selected one healthy long leaf. Okay, this healthy long leaf is to be inserted into the transparent bottle through the splitted cork. Before the inserting the long leaf in through the splitted cork, the bottle should have potassium hydroxide solution. This is the compulsory precaution. Before the inserting leaf through the splitted cork into the bottle should have 10 ml potassium hydroxide solution. So this is 10 ml potassium hydroxide solution or potassium hydroxide granules or solution, whichever may be, it has to be taken into the bottle. Then after, so like this, leaf is inserted through the splitted cork into the bottle. Before inserting, bottle should have potassium hydroxide. So like that, this arrangement should be made. Then after inserting the splitted cork, if it is any gap, this gap should be covered or covered with some Vaseline. For what? For further movement of air into the bottle should be prohibited. That's why the splitted cork, where there is splitted cork, that splitted cork part, if there is any empty space, that empty space should be covered with the Vaseline. So why we have taken the potassium hydroxide solution in the bottle? Because here potassium hydroxide is a good absorber of carbon dioxide. Potassium hydroxide absorbs total carbon dioxide from the bottle. Suppose if it is empty bottle, that empty bottle is with some air. In the air there might be present of some carbon dioxide. That carbon dioxide should not be present in the bottle. That's why here potassium hydroxide is taken into the bottle. For the good absorber of carbon dioxide, here potassium hydroxide is needed to be taken into the bottle. So potassium hydroxide absorbs all the carbon dioxide from the bottle, so that further no carbon dioxide is available in the bottle. That's why we have taken potassium hydroxide. So like this, leaf is inserted into the bottle through the splitted cork. Before inserting the leaf into the bottle, we should have taken potassium hydroxide. Potassium hydroxide is a good absorber of carbon dioxide, so that this leaf never take carbon dioxide or leaf never absorb carbon dioxide. So like this, if we wait for some time, after some time we can observe some things. So that this is all about procedure. Next, observation. Then after few hours, after 3 to 4 hours, see, after 3 to 4 hours, here what shall we observe? To observe the changes which have been made in the this experiment, here we have to be taken out the leaf which is inserted into the bottle. So carefully that leaf should be plucked out from the plant and detached the leaf from the plant and carefully taken out from the splitted cork from the bottle, and it is spread it over the petri dish. Okay, this leaf should be spread it over the petri dish. When we pour some iodine solution on this leaf, see here, when we pour some iodine solution on this leaf, so this is the leaf which we have done the experiment, no? So that leaf, it is spread it, the leaf should be spreaded on petri dish and pour the iodine solution. So this is the half of the leaf which is inserted into the bottle, and this is the half of the leaf outside of the bottle, and pour the iodine solution on both the sides, the leaf which is outside of the bottle and inside the bottle. On both the sides, if we pour some iodine solution, what happens? The leaf which is exposed to the sunlight, and the leaf which is outside of the bottle is turned into blue or black in color. Why the leaf which is inside the bottle is not turned? Even inside the bottle, leaf also absorbed the sunlight, but carbon dioxide is not present. Before absorbing the leaf, that carbon dioxide is everything consumed by the potassium hydroxide. That's why carbon dioxide is not available to the leaf. Only water is present, sunlight is absorbed, chlorophyll is available, but carbon dioxide is completely absent to the leaf which is inserted into the bottle. Because of the lack of only carbon dioxide, here the leaf which is inserted into the bottle is not performed any photosynthesis, so that the leaf which is inserted into the bottle is not shown any color change when we poured iodine solution on it. With this we have proved that carbon dioxide is necessary for photosynthesis. So this is all about carbon dioxide is essential for photosynthesis. Okay, just I will explain in the Telugu also. See, ఈ ఎక్స్పెరిమెంట్ చేయడానికి ఒక కుండీలో పెరుగుతున్న మొక్కను తీసుకొని కొద్ది రోజులు 3 నుంచి 4 రోజులు చీకట్లో పెట్టేస్తే దీంట్లో ఉన్న రిజర్వ్డ్ ఫుడ్ మెటీరియల్ అంతా వినియోగించబడుతుంది. కాబట్టి ప్లాంట్ అంతా ఇట్ ఈస్ కంప్లీట్లీ ఫ్రీ ఫ్రమ్ ద స్టార్చ్. అప్పుడు తీసుకొచ్చి ఎండలో పెట్టేసి ఒక పొడవాటి లీఫ్ని స్ప్లిటెడ్ కార్క్ బాటిల్లోకి ఇన్సర్ట్ చేయాలి. ఈ స్ప్లిటెడ్ కార్క్ ఉన్న స్ప్లిటెడ్ కార్క్ బాటిల్లోకి ఇన్సర్ట్ చేస్తే ఇది స్ప్లిటెడ్ కార్క్. ఈ స్ప్లిటెడ్ కార్క్లో నుండి హాఫ్ ఆఫ్ ద లీఫ్ ఇన్సర్టెడ్ ఇన్ టు ద బాటిల్, హాఫ్ ఆఫ్ ద లీఫ్ అవుట్ సైడ్ ఆఫ్ ద బాటిల్. ఇలా ఇన్సర్ట్ చేసిన తర్వాత ఈ లీఫ్ స్ప్లిటెడ్ కార్క్ పైన వ్యాజ్లిన్ పూసేయాలి, ఫర్దర్గా ఎయిర్ లోపలికి పోకుండా. మరి ఎందుకు ఇలా చేస్తున్నామంటే ఈ లీఫ్ ఇన్సర్ట్ చేయడానికంటే ముందే దీంట్లో బాటిల్లో పొటాషియం హైడ్రాక్సైడ్ సొల్యూషన్ తీసుకున్నాం. ఇది కార్బన్ డైఆక్సైడ్ని అబ్జార్ప్షన్ చేసుకునే క్యారెక్టర్ కలిగి ఉంది. కాబట్టి లీఫ్ ఎట్టి పరిస్థితుల్లో కార్బన్ డైఆక్సైడ్ తీసుకోదు, అబ్జార్ప్షన్ కాదు. కాబట్టి ఇన్సైడ్ లీఫ్కి ఎట్టి పరిస్థితుల్లో కార్బన్ డైఆక్సైడ్ అందదు, కానీ అవుట్ సైడ్ లీఫ్కి మాత్రం కార్బన్ డైఆక్సైడ్ ఉంది, సన్లైట్ ఉంది, వాటర్ ఉంది, క్లోరోఫిల్ ఉంది. బట్ లోపల స్ప్లిటెడ్ కార్క్ నుండి లోపల వైపు ఉన్న లీఫ్కి మాత్రం కార్బన్ డైఆక్సైడ్ అందదు. కాబట్టి మనం దీన్ని తర్వాత ఒకవేళ ఐరన్ సొల్యూషన్ వేసి టెస్ట్ చేసినట్లయితే ఈ లోపల వైపు లీఫ్ ఏదైతే ఉందో అది బ్లూ ఆర్ బ్లాక్ ఇన్ కలర్లోకి మారదు, అంటే స్టార్చ్ తయారవ్వదు. దీని ద్వారా మనం కార్బన్ డైఆక్సైడ్ అవసరమని ప్రయోగపూర్వకంగా నిరూపించవచ్చు. ఇప్పుడు కొన్ని ప్రికాషన్స్. సో వాట్ ప్రికాషన్స్ హ్యావ్ టు బీ టేకన్? ఫస్ట్, బిఫోర్ ద ఎక్స్పెరిమెంట్ ద ప్లాంట్ షుడ్ బి కెప్ట్ ఇన్ ద డార్క్ రూమ్. దిస్ is the first one. Second thing, while we are inserting the leaf through the splitted cork, the leaf, half of the leaf outside and half of the leaf must be inside. Third one, when we inserted the leaf into the transparent bottle, the leaf which is inserted into the bottle should not be touched to the walls of the bottle, because if it is touched to the walls of the bottle, there might be something change in the color. That's why leaf should not touch to the walls of the bottle. So these are the most important precautions that we have to be taken during this experiment. So this is all about carbon dioxide is essential for photosynthesis.